Hello, I am Professor Charu Dogra Rawat from Ramjas College, University of Delhi, and continuing with our ongoing series in biotechnology, today I will be talking about the production of cloned and transgenic animals. The learning objectives for the session would be to understand the need for the production of transgenic animals and to understand various methods of production of these animals. The references used in these sessions for educational purposes is T.A. Brown, The Gene Cloning and DNA Analysis, and Glick, Pasternak and Patton, 2010, Molecular Biotechnology, Principles of Applications of Recombinant DNA, otherwise stated in the particular text. Now, we have been discussing about biotechnology, and in animal biotechnology, we have covered the basic tools of genetic manipulation, in which we have talked in detail about the recombinant DNA technology, where we talked about the restriction endonucleases, the ligases, the transformation methods, and then we also talked about the construction and screening of genomic and cDNA libraries. Now, these basic tools of genetic manipulation are very essential for doing any kind of manipulations with the genome, which is the foundation for the animal biotechnology as we foresee. We also discussed about various advanced tools and techniques, in which we covered various blotting techniques, the northern, southern, western blotting. We talked about various methods for sequencing or knowing the base composition of DNA, in which we also covered the latest next generation sequencing methods. We talked about DNA microarray, and we have also covered the various gene editing tools covering the CRISPR-Cas9 system as well. All these lectures can be found on the CEC Gurukul site. Now, today we deal with the objective of genetic engineering is often to create a useful product or a commercial process. When we started with talking about animal biotechnology, now biotechnology is a technology which uses the biology of an organism. The purpose of this biotechnology or the recombinant DNA technology or the genetic engineering is basically to create some kind of a beneficial product or a commercial process which is useful for the, some kind of human intervention is there, which is again useful for the either humans, living organisms basically. It can be useful for living organisms, it can be human beings, it can be useful for various other animals, it can be useful for plants, but basically useful for the living organisms. One of the uses and applications of genetic engineering is the production of these cloned or transgenic animals. Now, we have talked about molecular cloning in detail. Just to review a little bit, the molecular cloning or recombinant DNA technology involves the generation of recombinant DNA. Now, this recombinant DNA is the DNA when the DNA from two different sources are combined together. And when they are put into a particular host, such as in bacteria or in bacteriophages or generally in bacteria, for making its own copies in large number, that is termed as molecular cloning. And when we are doing this deliberately to insert our gene of interest into an organism and looking at its expression or some kind of even involved in gene therapy, etc., then we say that it is the genetic engineering. So, we are creating or engineering the genomes. Now, the one application is the production of, as I said, transgenic animals. Now, what are these transgenic animals? A transgenic animal is one that has integrated a gene or DNA sequence, which is called as a transgene, which has been transferred by human intervention into the genome of a cell. So, of course, this is biotechnology because there is human intervention. There is a technology which is developed on the basis of biology, and the biology is generally that a gene can be communicated from one organism to another. So, this is a transgenic animal is the one that has got integrated a gene or a DNA sequence. Now, transgene because it is coming from another source into the genome of a cell. Now, the important point here to remember is that just the transfer of a gene or the integration of a gene represents cloning. Ideally, it basically represents just a recombinant DNA, not even cloning, because cloning means producing more copies of each other. So it basically means that a recombinant DNA is generated. So when you transfer, you can transfer it into a bacteria, but in this case, if you transfer it into an animal, then it is basically the recombinant DNA that you have created. So for more deeper discussion purposes, a transgenic animal is defined as one that has stably incorporated the transgene into its germline and is able to pass this transgene on to its offspring. So the stably incorporation the transgene into its germline is very important, because the recombinant DNA parts, when goes into molecular cloning, that you can produce the transgenic animals with desirable features, or you can express your gene of interest in the animals, and that is how it is called as a transgenic animal. So you are creating a breed of it. It has to be stably incorporated so that you get the offsprings with the desired characteristics as well. So that is an important point to remember about the transgenic. Just the integration of the transgene into the genome is one aspect, but it has to be passed on to the next generation as well. Now, before I go into the methodology of the transgenic animals, an important aspect is what is the need for the production of these transgenic animals? Why do we even produce them? Now, one application of creating more hybrid yielding strains is basically when earlier in farm animals, for example, if you want to produce efficient milk producing cow. So there are certain high quality cows which are present. So the selective crossbreedings are done so that the offsprings are also high milk yielding varieties. So these high yielding strains, even of different crop plants or farm animals, have been successfully developed by selective crossbreeding. So they may have more mass, if it is, for example, used for food, the animals, or they are milk yielding or egg laying capacities. So all these capacities are increased by creating these kind of hybrids by doing the selective crossbreeding, which is basically the animal husbandry that it involves. So these high yielding strains basically used to be produced by the selective crossbreeding. Now the problem is that this is a very time consuming process. Why? Because obviously it takes, there are a specific set of crossbreedings that need to be done, and every generation then the selection needs to be done, that whether you get those varieties or not, but you have to go through the entire process again and again. It's a very time consuming process. And it also requires improvements in product composition and production efficiency, especially in growth, disease resistance and reproduction. So just the desirable character is not enough. You also have to increase the efficiency of the reproduction in animals, or growth in animals, or for example disease resistance in animals. So all these kind of improvements are also needed. So what happens is that this transgenesis, which is the method of producing transgenic animals, it is a very powerful technique for studying the fundamental problems of mammalian gene expression and development, for establishing animal model systems for studying human diseases. So there are many different aspects into it. One thing is that we can, instead of using selective crossbreeding, which is still a very prevalent method, we can generate transgenic organisms of containing these high quality characteristics. Apart from that, the transgenesis is also involved if you want to express the mammalian genes for establishing these animal model systems for studying human diseases. So all the human, we have, when we started with biotechnology, we also talked about the importance of the model systems. Even if you are studying human diseases, you have model systems in mice, which represent those diseases, and then only you can, experiment on them and understand more about these systems. So for doing that, you need to express the genes of mammals in mice, for example, and that is done by this transgenesis. So they can be used for studying human diseases. They can be used for producing foreign proteins, for example in bird eggs, and for using the mammary gland to produce pharmaceutically important proteins in milk. The latter is called as farming, and we'll come back to all these when we talk about the applications of transgenic organisms. Today, the main focus is on the methodology and to understand what these animals are and what is the need for these creating or generating these animals. But we'll talk about the applications in our subsequent sessions, and then we will see that how these transgenic animals help in studying human diseases or for the production of foreign proteins or for using the mammary gland to produce these pharmaceutical important proteins in the milk. So, the production of the transgenic livestock, as I was saying, has the opportunity to significantly improve human health, enhance nutrition, protect the environment, increase animal welfare, and decrease the livestock disease. So, the advantages of these transgenic animals are manifold. So, not only they are giving us the model systems to study various kinds of diseases or to produce our desired protein products or to generate even the pharmaceutical products or the drugs etc. that we require, even the vaccines that we require in large quantity. It is not only helpful in that, but it is also helpful in increasing the animal welfare and decreasing the livestock diseases. So, all these applications of transgenesis reflects the significance of the methodology of transgenic organisms, and we will definitely talk about and take example of each one of them in our subsequent sessions when we talk about the applications of transgenic organisms. Now, coming back to how are transgenic animals produced. Now, apparently it's a very simple strategy. You have got a cloned gene. Now, I don't want to explain the cloned gene again, because if you have been following this series, you already understand what a cloned gene means. So, a gene that has been cloned into a vector, a recombinant vector. So, cloned gene is basically a recombinant DNA molecule that we are talking about. So, this cloned gene is injected into the nucleus of a fertilized egg. That is the first step, that you create a cloned gene in vitro and then you put it. So, injection is one of the methods, but you put it into the fertilized egg, nucleus of a fertilized egg. The second is inoculated fertilized eggs are then implanted into a receptive female for successful completion of mammalian embryonic development. Now, because we are containing transgenic animals, we understand that the embryonic development cannot happen outside the female body. So, we can fertilize the eggs in vitro, but for the embryonic development, we have to inoculate these fertilized eggs and implant them into a receptive female, which are also called as the foster mothers, for successful completion of the mammalian embryonic development. The third step would be then we observe the progeny or the offspring. Now, some of the offsprings derived from the implanted eggs carry the cloned gene in all of their cells. So, it is very important that why it is done at the embryonic stage and at what stage. So, generally it is done at the 8-cell embryo stage. So, when it is done at that stage, that is basically for even retroviral infections or nuclear transplantation, but it can also be transferred. There is a zygotic transfer as well, that it can be transferred into the zygote, directly into the nucleus of the zygote. And when these genes are expressed, then we observe the offspring and analyze them for the expression of the transgene. Once they are integrated in the germ cell lines and we identify the organism, which are called as the founder organisms, then they are bred to establish the new genetic lines. These are the new transgenic lines which are expressing the gene that you have been inserting, that you have inserted in the foster mother, in the basically the developing organism. So, it is the animal transgenic. So, this is a very simple strategy. You obtain the gene of interest in a particular way, then you introduce it into a embryo cell in again using certain methodology. Then you observe the offsprings or progeny and analyze to derive the eggs carrying the cloned gene, and then you get the animals with the cloned gene to bred themselves and to bred and establish the new genetic line. So this is the overall strategy of producing a transgenic. Now we'll look into the details of the transgenic mice methodology, and we'll be starting with the retroviral vector. So we'll just talk about that how this DNA can be introduced into the mice. We'll talk about the mice first, because there are various other transgenic animals that have been produced, and we will take certain historic or milestone experiments where this has been done, but as a generalized model we'll talk about the creation of the transgenic mice. Now DNA can be introduced into mice by retroviral vectors, which infects the cells of an early stage embryo prior to implantation into a receptive female. We'll talk in detail about what these retroviral vectors are and what is the mechanism of the introduction of these vectors. But one of the method is that you clone your gene in retroviral vectors, and then you make these vectors, or basically the viruses carrying these vectors, or the viruses act as the vectors and infect the cells of an early stage embryo prior to the implantation into a receptive female. The second method that we'll talk about is termed as the microinjection into the enlarged sperm nucleus or the male pronucleus of a fertilized egg. So in this case, the cloned DNA is directly injected into the male pronucleus of a fertilized egg. Or there is another third method in which you introduce the genetically engineered embryonic stem cells into an early stage developing embryo before implantation into a receptive female. So this genetically engineered embryonic stem cells we'll talk about. So there are certain stem cells. Stem cells are the cells which can give rise to any other cell of the body. So they are ever dividing cells, and they can give rise and differentiate into any of the cells depending upon the region, the conditions, etc. So at the embryonic stage you can have these stem cells, because at the embryonic stage the differentiation has not yet started. So these embryonic stem cells you genetically engineer. So genetically engineering again means that you employ recombinant DNA technology to create the recombinant vectors carrying your gene of interest, which is the transgene, and then you introduce these recombinant DNA into these embryonic stem cells, and then these embryonic stem cells, when develop into the, when get differentiated and give rise to the adult, then you get a transgenic animal which is there. So these are the three broad methods, and we'll take one by one each one of them. There are certain other methods as well. So we'll talk about the vector mediated gene transfer, in which we will basically talk about the retroviral vectors and lentiviruses today, and some other vectors we'll talk about in our later sessions. Then there is DNA microinjection, in which there is pronuclear microinjection, in which the direct recombinant DNA carrying our transgene is injected into the pronucleus of the fertilized egg, or there is embryonic stem cells where again the DNA, because we said that genetically engineered embryonic stem cells. So how would they be genetically engineered? You will create the recombinant DNA in the vitro, and then you will transfer this DNA, probably by microinjection as well, to embryonic stem cells, and these embryonic stem cells will then develop, and there is a transplantation of these stem cells as well, but these will develop into the they'll cause the expression of the transgene when the organism grows. Then there is some other method in which the gene transfer is directly into the gametes. So there is sperm mediated gene transfer, or there is testis mediated gene transfer. So we will also talk about these, where the genes, the transgenic vectors, are directly transferred into sperms, or they are transferred into testicular regions where the sperms are produced, or during at that time, so that the transgenesis can occur. And we will also talk about the somatic cell transfer, which is also called as a nuclear transplantation, where the somatic cell nucleus is basically transferred to another growing embryo, and therefore the transgenesis can occur. So these are the various methods, and we will start with the first method today, which is the vector mediated gene transfer. Now, talking about the vector mediated gene transfer method, there are certain retroviral vectors. Now, reviewing what the retroviruses are, the retroviruses have RNA genomes that are used as templates for reverse transcriptase to synthesize a DNA copy that can be inserted into the host cell genome. Now, we have talked about various viruses. I would also be starting, and I took some introductory lecture in microbiology. So, in microbiology, we talk about the bacteria, viruses, fungi, etc., the unicellular organisms, which are not the unicellular only, but we basically talk about the microorganisms. So, there we also talk about the retroviruses. So, there are certain DNA viruses and there are certain RNA viruses. These RNA viruses, they contain RNA genomes. Now, these RNA genomes, they can be used as templates for reverse transcriptase. So, I hope you remember what reverse transcriptase is. Reverse transcriptase is the enzyme which is causing the reverse of the transcription. So, what is transcription? In transcription, the DNA goes to RNA, but in reverse transcription, the RNA will go to DNA. So, there is a single RNA, single-stranded RNA, and the reverse transcriptase will catalyze the formation of a DNA copy. Now, this DNA copy then can be inserted into a host cell genome. We will talk about the detailed mechanism of these retroviral vectors. Now, virus-mediated gene delivery system. Now, when we said that these can be inserted into the host cell genome, what is the method by which these retroviruses, or as we would say, the retroviral vectors, because these retroviruses, when act as vectors to carry our transgenes, these are termed as retroviral vectors. So, how would these retroviral vectors mediate the gene transfer into the host cell genome? So, the virus-mediated gene delivery system uses the cell receptor recognition system of the virus for binding to a specific cell type, which is then followed by the internalization of the vector DNA and its transport to the cell nucleus. Now, again, this is very, very important, that how retroviruses are able to introduce the retroviral vectors into the host genome. So, they follow their normal infection routine methodology. So, basically, they recognize certain cell receptors. So, cell receptor recognition system of the virus. So, they bind to a specific cell type. Now, this specific cell type is again very important here, because if we want to do the expression in certain cell types, then also this system becomes very useful, because if we are introducing it into an embryo and the embryo becomes an adult, then generally it would be that every cell will contain a transgene. But they will contain a transgene, but if the expression needs to be regulated, or even when you are entering it there, then certain cell types, it will enter certain cell types only because they have got certain cell receptors. So, even when the transgenic product is produced in the cells or outside the cells, they do not express in all the cell types. They express in certain cell types only. So, the virus-mediated gene delivery system uses the cell receptor recognition system of the virus for binding to a specific cell type, which is followed by then the internalization. So, they will bind to the receptors present on the host cells, and then they will introduce their genome, which in this case would be RNA, or the vector DNA, whatever. If the reverse transcription has occurred and there is vector DNA, then it will introduce the vector DNA, and then it will be transported further to the cell nucleus. So, this is a generalized gene delivery method system usually for all the viruses, but we will talk about the retroviral vector system and its employment in the generation of transgenic mice in our subsequent session. Thank you.